Today is September 27th, 2023, and I will be reading Consensus Summary Report for CEPI, BC, March 12th through 13th, 2020. Meeting Assessment of Risk of Disease Enhancement with COVID-19 Vaccines. And I just want to go to um, some really important parts in here. I'm not going to read the whole article. And it says one of the challenges facing rapid vaccine development for SARS-CoV-2 is the need to adequately assure the safety of these vaccines. One such safety concern is disease enhancement syndrome that occurred in the 1960s with inactivated RSV and measles vaccines. Vaccine-mediated disease enhancement is characterized by a vaccine that results in increased disease severity if the subject is later infected by the natural virus. During early trials with inactivated RSV vaccine, the vaccine did not prevent infection. 80% of those infected required hospitalization, and 2 children died. Lung pathology in patients showed an unexpected inflammatory response with both neutrophils and eosinophils, evidence of immune complex formation and complement activation in small airways. Scientists later learned that the vaccine caused a similar disease enhancement in animals, characterized by immunopathology and a T helper cell type 2, Th2, bias response, and antibody responses with poor natural neutralizing activity. Since that time, the animal models have been relied upon to predict safety for new RSV vaccines that are developed. Of note, the pathogenesis of RSV disease enhancement is distinct from antibody disease enhancement, which occurs for macrophage trophic viruses, demonstrated most notably for dengue in humans and the coronavirus feline infectious peritonitis virus in cats, and is directly caused by non-neutralizing or sub-neutralizing antibodies, leading to more efficient viral uptake of via the FCY receptor binding. This is important right here. Since pathology consistent with RSV vaccine enhanced disease and perhaps ADE has been demonstrated for some SARS-CoV-1 vaccine candidates in animal models, there is also a concern that a similar syndrome could occur in humans immunized with SARS-CoV-2 candidate vaccine. Therefore, CEPI and the Brighteon Collaboration Safety Platform for Emergency Vaccine convened a safety working group. So, and then I wanted to go over here and share with you about, let's go to the very top, COVID-19 vaccine mediated enhanced disease and vaccine effectiveness and the vaccine data and the vaccine safety data link. And I'm going to skip through all of this. Well, let's just read this part right here. Okay. Vaccine-mediated enhanced disease can result from immune complex formation and Th2 biased cellular immune response when vaccinated individuals become infected with the target pathogen. Animal studies of candidate vaccines against SARS-CoV-1 have raised concerns about potential vaccine-mediated enhanced disease in recipients of COVID-19 vaccines. But I wanted to show you something else. I want to go here to the background. It says COVID-19 has created once in a lifetime public health crises. More than 60 million COVID-19 cases have been diagnosed in the US, and over 835,000 people have died from this disease. Vaccine development has occurred at a record pace, and three highly efficacious vaccines have received emergency use authorization by the Food and Drug Administration. Two vaccines use in messenger RNA platform with a lipid nanoparticle delivery system. The modified messenger RNA codes for the stabilized perfusion form of SARS-CoV-2 spike protein. The Moderna vaccine, messenger RNA 1273, is authorized for use in persons aged greater than 18 years. It is administered as a intramuscular dose at 0 and 28 days. The Pfizer BioNTech vaccine for persons aged greater than 12 years at whatever dose at 0 and 21 days for children aged 5 through 11 years. On August 23rd, 2021, it received FDA approval for use in persons aged 16 years. Individuals 5 to 15 years continue to receive the Pfizer BioNTech vaccine under EUA. Now, here's the most exciting part. The Janssen vaccine uses a replication incompetent human adenovirus AD26 vector to deliver a transgene for stabilized spike protein. A transgene. Well, what is a transgene? A transgene is an experimentally constructed piece of DNA that has integrated into the genome of a recipient organism. Once integrated into germ cells, subsequent generations inherit the transgene, referred to as stable transgene transmission. An experimentally constructed piece of DNA. I'll leave it there.